Okay, hello everyone, and thanks for tuning in. Big thanks, first of all, to Sammy and all at Biga for inviting me to do this talk today. And again, big congratulations for all the work you put into this week of education. Very well done. So it's Turfgrass Disease Control 2020 Vision is called today. And what am I going to be talking about? Well, I put this together back in January 2020, just after Harrogate, and it was for at least a 1 hour talk. But things changed, of course. So we're cut back to 30 minutes. So I'll have to skip through some of the stuff, but still, what I will be talking about is research and data regarding non-fungicidal disease control. I want to talk about Microdolcum and Anthracnose infection. Just a quick run through as to how they actually infect turf grasses. And I'll mention some cultural and environmental factors that we can actually influence to reduce disease pressures. Some scientific research supporting various tasks we can carry out. But the main focus would be on plant health and looking at ways to prime and stimulate various defense mechanisms in turf grasses. And because of that, we'll be looking at the use of nutritional programs and nutritional inputs, which can help to suppress disease. And for that, I'll actually be looking and showing some trial results over the last couple of years, and also looking at the current research I'm doing, which started last September. So that's the plan. We'll move on. So let's have a look at actually what we're dealing with first of all, okay? This is just some of the various pathogens and diseases that we deal with in Ireland and in the UK. Obviously, if you're in parts of Europe or in the States or Canada with different climatic conditions, you'd have a whole range of other problems to deal with. But these are the main ones we deal with. And of course, of these, the two major ones, I think you'll agree, would be anthracnose through the summer and microdolcum or fusarium patch, which we can get from September right through to April even, So these are the two main ones, and these are the two we'll be concentrating on today. So there's a nice green with some pretty devastating anthracnose visible on it. So what exactly is anthracnose? Well, it's a fungus. It's an Ascomycete fungus, and it causes two main problems in turfgrass. Two forms. You can have basal rot or foliar blight. Now, basal rot, as you can probably work out even if you didn't know anything about it, it affects the bottom or the base of the plants, the crowns, stem bases and roots. Mostly Poa annua, certainly it will affect other species, but Poa is very susceptible to it. And as you can see up in the photos here, I'll just turn on my little, here we go. You can see how it affects the base of the plants, this darkening, this reddish dark colour, and it has stunted root development and that, so it can be very devastating. I think the main form that we would be dealing with, though, would be the foliar blight of anthracnose. I'm sure everyone's very familiar with seeing this through the summer months. Mostly a summer problem, as I said. It affects the leaves and the shoots, discolors them. It can be, mistaken sometimes for drought stress, so you have to watch out for that. But again, this part actually attacks the stems and leaves mostly. So what can we say about it? Well, basically it would indicate a problem with your turf. So if you're getting anthracnose, it's very much a stress related problem. So we have to, if you don't want to use fungicides, or if you haven't got any available, you have to look at ways, first of all, of reducing stress on your turfgrass swards. A healthy plant is much more able to withstand infection and ward off anthracnose. So we have to help plants respond to stress. Let's have a quick look at the infection process of anthracnose. And here up the top left where my laser is, you can see these are the reproductive structures. Now these will get onto the leaf of the plant and they will germinate and grow out, form these structures here where my laser is. This is an aprosoria. And basically this actually bursts through the outer layer of the leaf and allows the pathogen to grow into the intercellular spaces, into the cells, where it will extract nutrients. It then emerges and forms these dark structures here. Actually, you can see these with a normal hand lens, and basically these will actually just produce the reproductive structures again. So the whole cycle of infection just sort of keeps on carrying on. We'll have a quick look now at Microdolcum infection. Again, this is another fungus, an Ascomycete fungus. Causes a fusarium patch or pink snow mold. You know, we don't get much snow over here, but certainly, for example, in Canada or parts of Europe, snow cover for numerous weeks or even a couple of months, this can develop and form what's called pink snow mold. It's the same fungus. We're more used to the fusarium causing the sort of foliar. You're familiar with all the patch infection of fusarium or Microdolcum. It's the most common pathogen in turfgrass in the UK and Ireland, and very important right through any of the temperate climates in the world. Now, we looked at the infection process in great detail over the last few years, and numerous, as you can see, greenhouse samples in pots and trays, and of course, being in Ireland, we had no shortage whatsoever of outdoor trial plots and greens to get samples from. So we had loads of samples to work with. And one process that we did use to look at the infection process was called fluorescent microscopy. This actually uses various stains which attach to these fungal cell walls. And then when you're using your microscope, which has a UV ultraviolet filter on it, these fungal structures, as you can see in these photos, become very clear. So we could use that process to look at the whole infection from the source of the inoculum right through as it enters the plant and exits and reproduces. So with Microdolcum, what we found was briefly this. As I said, the hyphae, or the source of infection, was either the hyphae, the individual strands of the fungus, or conidia, the reproductive spores. Here you can see the strands. The blue is actually strands of the hyphae within a sandy root zone. So it would be there almost all the time. It's in your root zone, in your thatch layer. And when environmental conditions are correct for it, it will grow from here up the base of the plant onto the leaves. And this here is a creeping bent leaf. You can see the mass of the fungal hyphae on it. So it grows onto the leaf. And with Microdolcum, if you remember, I mentioned anthracnose actually forms an aprosoria and bursts through the leaf into the plant. With Microdolcum, in almost all cases that we looked at, and on other people as well, the fungus, the pathogen actually enters via the stomata, the respiratory openings on the leaf. You can see the opening here. So the hyphae would enter there, and it would go through the inter and intracellular spaces, travels through the inside of the leaf into the cells, where it extracts nutrients. So basically, it's like everyone, once this lockdown is over, out on a Saturday night eating and drinking, and like most people then, once it's had its fill, it likes to reproduce. And it does this by expelling or extruding all these numerous reproductive structures, the conidia, thousands of them. And these are then spread by water, by wind, by foot, by golf machines. So they're spread, and then they can lie dormant, and then when conditions are again correct, they will germinate, and again the process of infection can continue. And there's some nice photos of Microdolcum infection on golf greens. So there are the problems that we're talking about today, anthracnose and Microdolcum. Now, the answers used to be, of course, we'd had things like carbendazine, chlorothalonil, leprodion, and propiconazole, and these were very effective fungicides at controlling disease right through the year. Of course, these are gone. Now, we still have a number of available fungicides, so there's quite a lot still you can choose from. And in fact, matter of fact, there's two new fungicides became available this year, 2020 anyway. Signature, which is a phosphonate or phosphide-based fungicide, and then there's this, which is Acernity. So, there is fungicides still available, so no need to panic fully just yet. But what other alternatives or options do we have to reduce disease levels if you don't want to use fungicides, or you're trying to reduce their use? Well, just remember as well, it's not just golf greenkeepers that are fighting disease. It's the whole range of groundsmen and loads of sports surfaces. A nice collection of photos there of the various surfaces that are dealing with disease. So what we can do as groundsmen or fungicides is, first of all, look at this pyramid, this disease pyramid. So you have your levels of disease, right? And there's 4 factors here which have to be present and working in tandem to create the amount of disease you have to deal with. And as turf managers, you have to look at this pyramid and see what we can actually influence, and this will help to reduce disease pressures. Now, like I said, the pathogens, certainly the Anthracnose Microdolcum, they're near enough present all the time, so there's not much you can do about them. Now, you can look at the host plant. You can look at changing to a less susceptible species, or less susceptible cultivars of whatever species you're using. We will be looking at the nutritional inputs for host plants in a minute, and this actually interacts with the time factor, which means that we're looking at giving the plant more time to respond to infection with various defense mechanisms. We'll look at that now shortly. But what you can look at as well, and what you can influence, of course, is the environmental factors for disease. And with Microdolcum, for example, you look at any possible environmental pressures that are there and see how you can reduce them. Open up your course if you can, get the light and air moving. And of course, one key environmental factor would be, of course, dew. Leaf wetness is crucial for disease development. And like I say, we haven't got the full time to go through all the data and facts, but the ICL with STRI have produced a lot of research looking at how dew removal will have a significant effect on reducing microdolcium, and there's products and means to carry out that at the moment. Again, if you can, look at the timely use of aerations and top dressing programs. Now, as an ex-course manager, I know there's a lot of pressure sometimes to put back your aeration programs from, say, summer up to end of September, October, and you have to, if you can, try to avoid that, because putting out lots of heavy top dressing in October, you're creating a perfect microclimate for microdolcium development. Again, look at incorporating more rolling into your program, because there is strong scientific evidence to show that rolling along with mowing will actually reduce not only microdolcium but anthracnose and dollar spot. So if you can incorporate more rolling into your program, you might be able to reduce the frequency of mowing or increase the height of cut, and by rolling you can still have the same playing qualities. Again, look at all the factors that microdolcium likes, basically, and see how you can actually affect them. Now, of course, there are nutritional inputs, which we're going to follow in more detail, but nutritional programs which include things like phosphate, sulfur, salicylic acid, seaweed, again, as sequentially applied programs, will have a significant effect on microdolcium. So environmental factors for anthracnose, for example, pretty much the same, except as I said, anthracnose indicates a stress problem with turf, and you're going back to basically greenkeeping 101, which we learned years ago. Aerate if you can, reduce stress and on your swards is one way to do it. But again, there's much scientific evidence to show that sequential light top dressing, increasing your height of cut and rolling On their own, they will actually have an effect on anthracnose, reducing it, but working together as a program, it will have a very significant effect on reducing disease levels. Again, crucial for anthracnose is inputs, spoon feeding, say, of nitrogen through the summer months. There's plenty of data to support that, and of course potassium levels have to be adequate also. So inputs of N and potassium will help again to reduce anthracnose, and again we'll be looking at some programs in a few moments. As I said, we'll be looking at some of these programs, including all these various compounds and elements here. So just to go back over that cultural thing that you can do, you know, I say much published data here. For example, this from ICL here showing the reduction of microdolcum using a dew suppressant. This is from Rutgers University looking at the effects of rolling. Again, rolling will reduce anthracnose, as it will microdolcum and dollar spot. Again, like I said, you know, regular top dressing will also reduce anthracnose. And what happens here, as you can see, is the regular top dressing, sand top dressing, actually protects the crowns and reduces stress to the plant. And again, mowing height over here, going from 2.8 to 3.6 will actually have a very big effect as well. So, you know, putting all these four things together, for example, in a program will help to reduce anthracnose while maintaining healthy and well, you know, nice playing surfaces. So I mentioned the infection process a few minutes ago of anthracnose and microdolcum. Like I said, the pathogen is there. When the environmental conditions are correct, they will grow into the plant. Anthracnose by bursting through and microdolcum by stomatal penetration. But what I didn't mention was that, you know, infected plants actually will respond with some very complex defense mechanisms. And obviously we haven't got time today to go through all this. I mean, you could talk about it for hours. Well, maybe I could. Maybe you wouldn't want to listen for hours, but I could talk about it for hours. But what we have to look at is things that, you know, first of all, the plant has to recognize it's being infected, and this is where elicitors come in. And then the short-term sort of damage control responses, which includes things like the hypersensitive response and programmed cell death. But it also involves production of numerous compounds for defense, mostly what's called phenolic compounds. And these would include, you know, antimicrobial compounds like phytoalexins. But eventually it should lead on to what's called a whole plant response, or systemic acquired resistance, or induced systemic resistance. And this is where parts of the plant which hadn't been infected are primed and are better able to respond then to future infections. So it's pretty much like the vaccination programs that are rolling out at the moment. You're priming the plants to respond to disease before it actually gets it. And of course, for plants to actually produce these defense compounds, first of all, they need the resources. So if they're very badly undernourished or on a knife edge, they're going to be much more susceptible. So you need a balanced nutritional program, first of all, to ensure that the turfgrass actually has the means to produce these defenses. And then there's many elements and compounds that would directly stimulate or enhance these defense mechanisms. You know, as we saw with anthracnose, nitrogen and potassium are crucial to help reduce that. But, you know, things like iron, calcium is crucial because of the first line of defense involves what's called an ion flux, which involves calcium at the molecular level. So calcium can actually become very tied up. So available calcium is crucial for defense. Salicylic acid is another compound. This is a signaling compound which travels through the plant and kickstarts the defenses in other parts of uninfected areas. Phosphate, there's much evidence now to show that phosphate actually primes or enhances these defense mechanisms prior to infection also. Seaweed can be very beneficial also, as can things like glycine betaine. Chitosan or chitinase, what this is the fungal cell walls are made up mostly of chitin, and by applying or including, say, chitosan or chitinase in your programs, it actually tricks the plant. It picks up on the chitin and it thinks the fungus is actually invading or infecting, and it will stimulate these defenses. And also pigments. Also, there's evidence to show that using pigments can also enhance your plant health and again help the plant respond more rapidly to infection. So my research aims over the last few years are basically looking at nutritional programs to enhance plant health, but also to stimulate and enhance these defense mechanisms and thereby suppress disease. So that's the basic aims of the research I've been doing for many years now. And it goes back to 2007. This was one of the first things I did. Actually, it is the first thing I did looking at disease. And this was with Dr. Andy Owen at Myerscough, who's now with ICL and doing some great work looking at nutritional inputs for disease control as well. So this was done with Andy, and what we found was phosphate treatments to the greenhouse plants significantly reduced microdolcum. This was creeping bent back in 2007, 2008. Then we moved on and we had 4 years of trials, again briefly looking at the use of phosphate to control microdolcum in different species. And over the 4 years, we found that phosphate treatment would actually reduce microdolcum by about 50% compared to the untreated controls. Now, but I want to concentrate mostly actually on some recent work I've been doing. And I want to show trials from 2018 for anthracnose and from 2018-2019 with microdolcum. And then I want to show the current trials which started last September. And these including compounds such as NPK, phosphate, calcium, all these here which I mentioned previously. So we'll start with this Anthracnose trial here from 2018. This ran from July to October 2018. If you might remember, 2018 was a very hot, dry summer, very stressful for turf and for greenkeepers alike. So this trial ran from July to October. Treatments were applied every 14 days, and we looked at the levels of anthracnose. So there's 4 treatments went out basically. There was an untreated control. We had phosphate on its own, and then we had this nutritional program, which I'll give more detail on in a moment. And we had Banner Max, if anyone remembers Banner Max, as a fungicide control. So 4 treatments going out every 2 weeks. And this is what we found. This was the peak of anthracnose back in August 2018. As you can see, the control level was up at 35%, 36%, quite high. The phosphate on its own was very effective, reduced anthracnose down to 11%. But what I found most interesting was that this program of nutritional inputs was statistically the same as Banner Max. So very good control just by using various nutritional inputs there. And over the full trial period from July to September, you can see, the level of disease on the control plots was 20% overall. Phosphate reduced it down to 6.6. And again, the nutritional inputs on a par with the fungicide. So what was happening there was that, the phosphate, as we already knew from previous work, was actually priming and enhancing the turfgrass prior to infection. But the nutritional program we had, first of all, it was providing adequate nitrogen, potassium and phosphorus for that time of year. As like I said, nitrogen and potassium levels are crucial to reducing anthracnose. But we also had components in it that included sulfur, copper, there's iron, phosphate in it, manganese and zinc. And these are all crucial for priming these defense mechanisms. We also had a surfactant in the program, which first of all was reducing water stress and ensuring adequate infiltration and water management. But also it has an acid component in it, which was actually solubilizing the calcium in the root zone and making it available. And like I said, calcium is crucial for initial defense mechanisms. We also had salicylic acid. Again, as I said, this is a signaling compound which would stimulate this systemic acquired resistance. So overall there, this combination of products will actually lead into less stress. First of all, less stress equals less anthracnose, and they provided elements which data already shows that they will reduce disease incidence, and some of them are also priming and enhancing the plant's natural defenses. So the next trial I want to look at, mention, is the Microdolcum trial from 2018 and 2019. And again, this went in two parts really. We had a range of treatments going from September to January, every 14 days, and we looked at disease. And what we had again was a control. And again, we had a nutritional package here, and we had Instrata. I think Instrata is gone now as well, isn't it? So that's from September to January, and then from January to March, we had the same program, except we also included a 4.5% chelated iron. So we just fiddle around a bit with the inputs there. That's a scientific term, by the way, fiddling around. So what we got, this is what we got. This is the first section from October to January. Here's the control levels here, quite high. Again, the phosphate on its own was very effective. And the nutritional inputs were quite good also. Not as good as the Instrata, as you probably would expect, and statistically the same actually as the phosphate on its own. So interestingly enough, when we included the chelated iron in then from January to March, we had a further reduction in this for this nutritional package. So statistically it was a greater suppression compared to the phosphate on its own. So again, some good results there. Just a few photos of that from that particular trial in 2018-2019. So what was going on again? The phosphate was priming the defenses, and that nutritional package we were using, first of all, was ensuring adequate nutrition for that time of year, which was autumn, winter, so low inputs of nitrogen. But it had calcium supplied and phosphate for defense, and also salicylic acid. And like you saw, the chelated iron further reduced the disease levels. So those few programs there sort of spurred me on, and we put together a program earlier this year. I wanted to look at all these elements and compounds in various concentrations and various combinations. So we came up with this program, which started last September. And basically it's, there's 6 treatment packages here. And not only that, they are going out at either a 2-week or a 28-day, 4-week interval at different rates. So basically there's 12 treatment combinations going out and controls. And they're going in 2 sites. I have one at my own site here at home, which is a POA based on a sand-based root zone, and the second site is at the Seaweed Driving Centre in Dublin, which is POA, but it's an established green on a push-up soil-based root zone. And we're collecting a lot of data, as you can see. There's 13 different treatments going out if you count the untreated control, and we're looking at soil moistures, and we're looking at colour density, turf quality, NDVI readings, and of course, disease levels. And that was from the 1st of December. Now, already this is taken in December, just before Christmas, and we get some really good visual results, as you can see already. That's this one here actually is one of the treatment, number 6, which actually only has 2 products in it compared to the control. So very excited so far. Let me show you some of these results that we're getting at the moment. So this was taken November, a few weeks into the program, so disease levels were low enough. And as you can see, what we have to do here, the analysis and the interpretation of these results is going to be tricky enough because there's so many of them and so many different interactions going on. As an example, this one here, number 6, statistically it's the same as this one here, but different from that. Whereas this one here is the same as that, but different from this. So a lot of work will have to be done to figure out the best programs and the best treatment combinations and frequencies of application to get not only disease suppression but also good plant health and turf quality. But it's looking good so far. This is a more recent one from the 14th of December. I haven't done any statistical analysis on this yet, but you can see good level of disease on the control plots here. All the treatments are reducing disease, but these here, the last couple along here now, are very effective and very good with regards to enhancing turf quality also, which is a good combination, I can tell you. Like I say, it'll take a while to get the full benefit and full analysis done, but so just watch this space because we'll keep you updated as to what's going on. Again, like I said, turf quality is also important. As you can see, there is an enhancement of quality from all the treatments as well compared to the control. As you can see, the treatments have visually some quite a few differences in colour and that. Again, with regard to disease, some quite good pictures coming through. That's number 7, plot number 4. Some nice quality coming through as well from the treatments, I have to say. It's hard to tell sometimes from a photo. Again, disease levels very encouraging, I have to say. Another couple of photos there. And 2 more photos to finish with. You can see the difference in colour and disease between the 2 of them here. So we're getting near the end of our time. Actually, you're probably gone over it. So what's the take-home message from today's talk? Well, we need to look at the disease pyramid and see what factors we can actually influence to reduce disease pressures. And with regard to microdolcum, look at all environmental pressures such as dew removal, timely operations of aeration, incorporate rolling, and the nutritional programs to reduce disease. Anthracnose, reduce stress levels, incorporate light top dressing, increase height of cut, rolling, low inputs of N and potassium. And with regards to nutritional IPM, we've seen that from the various trials that all these compounds and elements combined or on their own will have an effect on disease levels, not only with microdolcum but with anthracnose. And of course, just don't take my word on it. There's plenty of data available out there. For example, over in Oregon State, Dr. Clint Mattox has done a huge amount of work over the last few years. And you can see here, for example, compare the control panel on the left to the one with phosphate and pigments and others inputs. A very good success at reducing disease there. And of course in the UK you have people like Headland who've been researching this area for many years with great success. I know their 2030 program is very good. Again with ICL with Dr. Andy Owen and Henry Bechelet. Huge amount of work going on there at the moment, looking at various nutritional inputs, which is also successful at controlling disease and in ensuring plant health. So have a look at all them. And that's about it, I'm afraid. So if you found it of interest, hopefully you did. I want to say thank you very much. And I'm on Twitter here, or there's my email on the left. Any questions or comments, don't hesitate. I'm always available for talking about stuff. So again, thanks very much for listening.